All right. So this is our final update for our cell type clustering with our autoencoder neural network. So our motivation problem statement. So essentially the data that our model is, the data that we're using for our model is coming from single cell RNA sequencing. This is essentially a bioinformatics data set that essentially looks at gene expression at the cellular resolution level. And it's a popular tool data set that scientists have used in bioinformatics to essentially group cells together, like making clusters or even inferring their cell type based on expression, differential expression of biomarkers. So our model is essentially taking that and making a deep learning approach to essentially can we classify certain groups of cells, based on their gene expression using a deep learning, more of a deep learning approach. Our data set's coming from an online database source, this mouse cortical brain development data set. Essentially, these mouse brain, they were taken at various time points. The RNA sequencing was done. We're only going to be looking at one time point to mitigate sample bias and to make sure that, when we're making sure our clusters are being made, that's not having an effect on that model. The data set we're working with is essentially going to be in a matrix format. So cells are going to be our columns and then the genes are going to be our rows. Um, it's not really a huge data set in the entirety of single cell data, since that could be in the millions. The gene expression, so like the data can just be like raw counts, which is like discrete. So just the number of times the gene appears in that experiment. And essentially the output in our model would just be the cluster label that a cell is assigned based on its gene expression. And because our data set, essentially because the model that we're using is essentially going through an unsupervised learning approach, we're going to have to be using a ground truth label. So the data set we're working with has ground truth labels that they have in the data set. So we're going to be using that to train our model so we can effectively improve and get a good idea of cells clustering, all that. And the two metrics that we're going to talk about are ARI and
Yeah, so RAND index, which is the non-adjusted version of ARI, is a count of like the number of correct sample pairs. So it essentially measures whether any two cells should be found in the same cluster or a different cluster. Again, adjusted RAND index just accounts for random chance in any clusters. Next. So mutual information measures how much information about one cluster tells you about another cluster. So for predicted clustering, essentially tells you how much information that gives you about the ground truth clustering. Adjusted mutual information also accounts for random chance. So putting these two metrics together, our baseline autoencoder model that used a mean squared error as its loss function, we used Optune to perform some hyperparameter selection for this model. It had an AMI of about 0.53 and an ARI of about 0.39. So it's like not great, but it definitely was learning something. So for these two graphs, the groupings of the colors are more important than like the colors themselves. So for example, like the red clusters in the top left versus like the light orange cluster in the top right, that means like the model predicted pretty well this cluster. But again, the performance wasn't great overall, so we ended up switching loss functions to improve performance.
So yeah, the new model architecture has some key differences from the standard autoencoder. Mainly these loss functions, these custom loss functions. Now essentially the model's learning latent features by reconstructing the input as a zero inflated negative binomial distribution, while also improving the cluster assignments with the clustering loss term. Next. So the reason the MSE reconstruction loss isn't ideal is that single cell count data is usually over dispersed, meaning it has a higher variance than the mean. It also usually has excess number of zero counts due to like sequencing errors where expressed genes were like not detected even though they were actually expressed. Next. Um, so the output of the decoder is now going to be the three parameters of the Zen-B distribution, which are mean, dispersion, and the probability that if the output is a zero, that it's from dropout, or if it's a real zero. So using those parameters, the loss function is now just the negative log likelihood of the ZinB distribution. Next. Um, and then the clustering loss function is essentially just a KL divergence between the predicted cluster distribution and like a target distribution, which is derived from the predicted one. This is essentially just sharpening the clusters, making them more well defined. Next. Um, yeah, so then altogether the whole loss, the loss for the whole network is a combination of the ZinB loss and the clustering loss. Next.
So overall, adding essentially these metrics and losses together for our model, we were able to improve the AMI and ARI of the autoencoder. Like Evan was saying, the colors don't matter. It's more the cells that are assigned to the cluster that matter. So you can see that while there are some irregularities, like there are still some cells that are misclassified as which cluster. What matters more is essentially the accuracy of that cell, and the overall scope in a larger data set. So you can see that here. And no prior work was done on this project, and here are the references. Yeah.